Okay, so I will give you a short introduction in the project SolveRD. We are working on this project and have started from January 2018. And this project will run until the end of 2022, so 5 years project. There are 22 partners from 10 countries, and it's coordinated by Olaf Ries and Holm Gresna in Tübingen, and co-coordinated by Han Brunner in Nijmegen in Netherlands and Anthony Brooks from Leicester. This is a slide about the SolveRD infrastructure. So it is it's working with 4 European reference networks, actually also 10, but 4 are building the core of SolveRD. Those are RD, EuroNMD, Ithaca, and Genturis. And we have also 6 associated networks and 3 undiagnosed patient programs from Italy and Spain. We also use further rare diseases infrastructure as the RD Connect platform, Orphanet, HPO, Eurogen Test, and the Canadian Models and Mechanisms Network. And we are also collaborating with 2 patient organizations, Eurordis and the Genetic Alliance UK. So the SolveRD project is dealing with 4 major cohorts. The first one is the unsolved cohort. So we are trying to gather 19,000 in total unsolved exomes and genomes, and in a standardized way, and to reanalyze them with the goal to solve many of them. The second cohort is the specific cohort, and it's defined that those cases have already an exome and will get further omics analysis, as well as they are determined by the 4 core ERNs. The third cohort, which SolveRD is dealing with, is the ultra-rare cohort, and those are phenotypically remarkable and peculiar patients with a rare disease and that have no exome until now. So there we will perform phenotype jamborees and exome analysis. And the 4th cohort is the unsolvables, and those are highly recognizable clinically defined diseases or syndromes for which no disease gene has been identified yet. So there we will also use all omics tools to crack them and solve several of them. So for more information, you can always visit the SolveRD website and follow us on Twitter. If you have questions, please ask in the chat. And now I will give over to Steve from SINAC in Barcelona.
So I'm Stephen Laurie. You hear me okay?
Yes.
Yes.
Okay. So I'm Stephen Laurie. I'm a postdoc in the data analysis team here at SINAG in Barcelona. I'm joined by Ricky Joshi and Leslie Matalonga and Carlos Garcia. And today the purpose of this webinar is to show you how to analyze your whole exome and whole genome sequencing data using the RD Connect GPAP, as we call it, the Genome-Phenome analysis platform. So this is the overview of what we'll try to get through. I'll go slightly more quickly maybe because we're a bit shorter of time than we anticipated. But basically I'll review the data workflow. I'll tell you a little bit about the RD Connect project. I'll tell you about the GPAP itself and how the GPAP allows you to share data. That will take about 15 minutes. And then I'll give you a walkthrough, a live walkthrough using the playground feature of the GPAP to show you how you would solve cases and highlight some of the advanced features. And then we'll take the questions at the end. As Sanya mentioned, if you can put questions in the chat box, send to everyone so everybody can see each other's questions. So we don't need to have any duplicates, and then we'll deal with them in the last 10 minutes, hopefully. So first, I'm just going to review a little bit what Leslie covered on Wednesday. So we had another seminar on Wednesday, and in case you missed it, this is what was covered by Leslie. So at some point, we'll be hosting that, I guess, on the SolveRD website, and you'll be able to access it then if you weren't here on Wednesday. So basically, this is the RD Connect homepage, the platform homepage. And most of what I'll be talking about today is within this red box in the center here. Leslie dealt with the registration, which is on the left, and the PhenoTips that is on the right in her talk on Wednesday. So to get your data into, so for those of you who weren't here on Wednesday, just to remind you how you get your data into RD Connect, or if you're a SolveRD partner, how you need to get your data shared into RD Connect as part of SolveRD. The first thing you need to do is register as a group under your PI. Then you will submit the phenotypic data using which we store in PhenoTips. Then you have to submit the metadata that describes your genomic data and transfer your genomic data to us, which are FASTQs or BAM files. And then once that's all done and processed through the standard analysis pipeline, we make it available to you again, and you can analyze your data in the GPAP, which is what I'm going to show you today. So I'm just highlighting here, there's a login button, there's a tryout button for the playground, and there's a frequently asked questions link. As I said, questions will be addressed at the end of the webinar. Please write them in the chat box in Zoom, and we will come to them then. So, the RD Connect project, just in case any of you, or for those of you who aren't familiar with it, very briefly. RD Connect was a 6-year EU-funded project, 2012 to 2018, and the goal of the project was to build an infrastructure for rare disease research. And the outcome is an integrated platform connecting data and clinical information, patient registries and biobanks for rare disease research. And what we managed to achieve was we created a central system for the reprocessing, storing, analysis and sharing of omics data, and including, very importantly, the integration of phenotypic data and biosample data. And another initial goal of the project was to contribute to the RD objectives of delivering 200 new therapies and the means to diagnose most rare diseases of genetic etiology by 2020. And one of the measures of the success of the project actually was that as of 2018, RD Connect, the GPAP itself, which we're talking about today, is an RD recognized resource. So this is something that we led on here in CNAG. We led the work package that was the development of the platform itself within RD Connect. So we've been involved with the GPAP. We've been in charge of the GPAP from the start, and now the GPAP is being used within SolveRD. So just a little bit of technicality for people who would be interested. So how do we process the data? What do we do? So the RD Connect, we have a standardized RD Connect variant calling pipeline that we've actually published. I haven't given the reference here. But basically we take a sample at a time. We accept FASTQs or BAMs. If you give us BAMs, we make them back into FASTQs. And then this is just a very standard variant calling pipeline used by many centers around the world, essentially mostly following GATK best practices. So we perform all these QC, trimming, mapping, duplicate removal, indel realignment, DQSR. The alignment's done with BWA-MEM, and variant calling done with GATK haplotype caller. And then we annotate using DBNSFP, Ensemble VEP, and currently we're still using the GRCH37 genome. And this generates a GVCF per sample. I then for each data release, we make a multi-sample VCF, and I pass that to the engineering team who have built the sophisticated big data backend to this process. So I pass them the multi-sample VCF, it goes into a Hadoop file system, it gets reformatted in Parquet, then extracted, transformed and loaded into Elasticsearch. And this is what allows us to make real-time queries on these big data sets. So if you think about an exome, it has somewhere between 50,000 to 100,000 variants typically, and a genome has somewhere between 3.5 and 4.5 million, and we want to be able to query these in a matter of seconds. And this is what the Elasticsearch allows us to do. And then we have a front-end user-friendly interface, which is what I'm going to show you today. And this was led by Davide Piscia and his team here in CNAG. So just to introduce you to the GPAP itself, before I actually log in and show you how to use it. So when you first log into the Genome-Phenome analysis platform, this is what you see. The current way we have it set up is that there's 3 sections. The first section is where you define the samples you wish to analyze. The second section is where you define the filters that you wish to apply to reduce your list of variants to a manageable set, or to try to identify the causative variant. And the third section is where you will see your results. So dealing with the sample section first. The first thing you do is you select your samples. So here, this is the same trio I'm actually going to use in the first part of the demo. So the case is the first sample, case 1C. We can also see the PhenoTips ID there. And there's the father and the mother, 1F father, 1M mother. And you define the genotypes by, you define the mode of inheritance you are interested by defining the genotypes. The reason we do it this way is because we've made the platform fully flexible, so you can do any sort of analysis on any number of samples from 1 to, it works with at least 15. We haven't got any pedigrees bigger than 15 individuals, and you can define the genotypes for all the individuals independently. So this is the mode of inheritance, and I'll show you how we do that in the demo. And then we also have some quality control parameters here. So we have the minimum read depth. Obviously, the more reads that support a variant, the more likely the variant is to be bona fide, because we should be aware that you always get some false positives, a relatively low fraction, but there's always some. We also have genotype quality, which is a standard quality measure. And then important filters that we have here also are the minimum alternate allele frequency and the maximum alternate allele frequency. So basically, if you're looking for a heterozygote, ideally, this value would be 0.5. In practice, heterozygotes with values between about 0.2 and 0.8 tend to be reliable. For homozygotes, you would expect the value to be close to be 1 or very close to 1 if it's homozygous alternative, or 0 or very, very close to 0 if it's homozygous reference. I say very close because we know there's always a few sequencing errors or a few alignment issues, and you might have a very low frequency of reads that are mismapped. So these values help you define the high quality variants first, which is what you want to look at. So moving on to the filters section. So we have 5 filter classes at the moment, shown here. So you can filter by the type of variant, you can filter by the control population allele frequencies, which is very important and very useful for rare diseases. You can filter using the SNV effect prediction tools. These are SIFT and Polyfem, Mutation Taster, CAD. Note that they, in general, only work for single nucleotide variants and not for indels. So we don't actually recommend filtering by them, but they are there as an option. You can filter by gene lists and phenotypic descriptions, which I'll show you in more detail. And also, if you're just interested in the variant in a specific position of a specific chromosome, then you can do that. Or you can limit your variants to runs of homozygosity, which I'll address again in the demo in more detail. So just to give you an example of the first one, so this is the variant type menu. You can see, first of all, you can filter by variant class. You can see there's a little black box here that's actually come up automatically. This is a tooltip, so when you hover your mouse over variant class, it tells you what variant class means. And many of the other fields, if you hover your mouse for a couple of seconds, you'll get a tooltip. So if you hover over high here, it'll explain what is meant by high. Likewise for moderate. And many other fields in the platform have these tooltips. So here you can filter by the variant class. You can filter by variants that are known to ClinVar, described in ClinVar. You can filter by protein coding variants or not. So when you're applying a filter, what you're doing is always restricting your maximum data set to a subset. So if you don't apply any filters, you get all the variants back. As soon as you start to click on these boxes, you get only variants back that match the filter you've applied. Another one we have is this gene and phenotypes filter. Here you can look for variants in your favorite gene just by typing the gene in and selecting it. We have some selected, some predefined gene lists that have come from either from ourselves at CNAG because we think they're useful for rare diseases, or requested by collaborators who are using the platform. I'll show those later. And also we can generate gene lists on the fly using the HPO terms that have been associated to the phenotype of the case in PhenoTips, which I'll show you. And likewise for OMIM. And also you can perform a union or an intersection of these. And another thing that's actually part of the standard analysis pipeline that I didn't mention in the single slide because it was cluttered enough, is that we also, for all samples that are submitted, we run analyses to detect long runs of homozygosity. Actually, any run of homozygosity in the sample, in the experiment, that appears to be more than half a megabase in length. And this is particularly useful when you've got consanguineous pedigree, because then it is most likely that the causative variant is going to be in the bit that's shared, which is between the parents. That is a run of homozygosity that they've inherited because they are consanguineous. And finally, once you've applied the filters, and you can apply as many or as few as you want at any stage, you can add or remove at any stage, then you will look at your results. And the results are shown at the bottom, and they look something like this. So the bottom half of the screen here is what we call the main results section, and the top half, the smaller part, the top third, let's say, is the extended results section. So typically, first you look in the main results section. Here you can see that this query, whatever it was, resulted in 11 variants being shown. You can see the chromosome, the position, the reference allele, the alternative allele, the genotype in the samples. You can see the gene name, the impact effect, high, moderate, low, and we have these color-coded effect prediction measures: SIFT, polyphen, mutation taster, CAD. Again, it's important to remember that these are only indications that these alleles might be damaging or disease-causing. They're not definitive. So these are machine-trained and they're not perfect. And what we're showing here is that there's at the position level and at the gene level, we have a large number of links out to all your favorite genomic resources, genome and disease resources. And if we don't have your favorite one, let us know, and we'll add it. So you can see here from the position, you can link straight out to Nomad or UCSC or Ensemble. And this means you don't have to go and query and log into, go and open Ensemble in your browser and query. You can just click right here and it'll take you to exactly this position in the database. And likewise for gene name, if you have an HGMD account, for example, this will take you to the entry in HGMD for this gene, or OMIM or Ensemble. So we're making the follow-up analysis as easy as possible. And also at the top here, I'm showing we have a link out to the Human Splice Finder, which is a useful tool at the transcript level if you have any putative splice variants that shows you whether they're likely to be damaging or not, which was developed by one of our partners in RD Connect in University Aix-Marseille, led by Christophe Beroud and David Salgado. And then once you have variants that you're interested in, if you click on a specific variant, so here I've just clicked on the first variant, which is chromosome 1, position 91,859,888. And then we have in the tab above, we have, or in the menu above, we have all these separate tabs. At the moment, we're just seeing the functional tab. And you can see the gene name, which transcripts are actually affected, the effect impact at transcript level, because what we show below is the effect impact, the maximum effect impact at transcript level. So it might not be, although it's a high impact here, it might not be high impact on your transcript of interest. For example, if it's a nonsense-mediated decay transcript and it's a high impact, it's probably not of interest for your disease, because it's unlikely that that transcript is functional. And then we have a list of other fields, some of which I'll show during the demo. And another tool that's useful that we have fully integrated is Exomizer, which was developed by Damien Smedley and Peter Robinson and their teams. And what Exomizer can do is for known diseases, it can associate a short list of variants. Well, it can take a short list of variants and a list of HPO terms shown here that is automatically taken in from the PhenoTips instance in RD Connect, and it can prioritize those variants based upon known relationships between the HPO term and diseases. So this doesn't help you find new disease genes, but it does help you re-find known disease genes that you might have missed or might have been unaware of, because these things are being updated all the time as well. So the final bit of this PowerPoint presentation is how the GPAP allows users to share data. So this is one of the most important features within rare diseases, is to encourage the people responsible for the samples to share their data, because by sharing your data, if you have an unsolved case, you might find other unsolved cases that are very similar, and then you have much more power to try to identify the gene, or even sometimes the variant that is causing that is responsible for the phenotype that's observed, that you would not find if you did not share your data. So RD Connect enables full data sharing and analysis within RD Connect for registered users. So I should stress here that RD Connect is free to join. Any bona fide researcher can get access. It's free to submit your data. Within SolveRD, you have to go through the SolveRD process, but actually you can, if you're not a SolveRD partner, and you're not going to be for whatever reason, if you're not linked to an ERN, you can submit data to RD Connect separately to get it into the GPAP. But we do control the users. We make sure you're bona fide. Leslie explained in the initial talk, we check the person is real, so nobody gets access who isn't a real researcher. And then they can see all the data at some point, because everything becomes post-embargo after a certain amount of time. We allow for partial data sharing outside RD Connect, obviously very controlled, meeting all privacy issues, GDPR compliant, etc. We're very careful with our privacy issues. A core requirement of having any genomic data in RD Connect is that you have to submit the corresponding phenotypic descriptions, because that's what gives the system extra power. And if you share your data, you are more likely to solve your cases through internal or external matchmaking. And we're involved in initiatives such as Matchmaker Exchange that you may have heard of. So Matchmaker Exchange is a number of organizations that are performing similar types of analyses, trying to solve rare disease cases. And what you can do is you can send out a query and ask these other institutions, do they have a sample that looks like mine that has similar variants, or maybe has a gene that might be harboring variants of interest? And the answer is no or yes. And if you get a yes and it looks good, then we can put you both the groups in contact with each other. And we're also involved in GA4GH Beacon. I won't go into that in more detail here due to lack of time. So these are the main people to thank here at CNAG. You can see we're quite a big team now, and many members of CNAG who are not direct members of RD Connect or SolveRD have also play a role in various steps of the pipelines here. And this is the main groups that are involved in SolveRD at the moment, which is coordinated by Olaf and Holm in Tübingen, and Sanja and Berta are doing a very good job coordinating. So now I'm going to move to a live demo. So I'm going to walk through a couple of illustrative cases, I guess, and then I'll briefly touch on some advanced features of the RD Connect GPAP, which separate it from other tools, and most of the other similar tools you have to pay to get access to. So not only are we better, but we're also free. If you heard somebody laughing, that was in my team. So, okay, so let me go to where I need to go to choose Chrome, I believe. Oh, okay. Are we in the right place here? No, we're in the right place here. Okay. So here what I'm using is some examples that we generated. So these are pseudo-real cases. So what we actually took was we took a HapMap trio, whole genome sequencing data. If you're familiar with these things, it's the Illumina Platinum trio. The case individual is NA12878, and we have her mother and father. And then what we did was we took real variants that have been identified in RD Connect for trios, and we inserted them into this data. So this is to keep it anonymous, but to show you that it works with real data, right? So this is a whole genome sequencing trio. And within the platform, actually, within the playground, what we did is we used this same trio 9 times now. So it's the same data sets for the different cases. The only thing that's different between them is the variant, the causative variant that we have inserted. So I realize there's something else I meant to show here. Sorry, just give me a second. Something I forgot to preload. here you can see the case description of the first case I'm going to look at. It's called case 1. The case is 1C. Do you see my mouse?
Yes.
You do?
Yeah.
Okay. So case 1C and his parents, because a boy in this case, father and mother. So this is sort of a summary of the type of data that we get, phenotypic data, right? So this individual is 5 years old when presented. Looks like it has congenital myasthenic syndrome, CMS. And here are the main clinical features: neonatal hypotonia, distal arthrogryposis, an inability to walk, and recurrent lower respiratory tract infections. Okay, so now we're going to load these samples into the playground. So here I am in the sample selection. Here's the view I showed you before. So I'm going to load 3 samples. I'm going to use the case first. So this is case 1C. You'll note that when I select, the background turns green. If the background does not turn green, then the sample has not been correctly selected, right? Okay, so looking at this case, it's unclear. We can't be sure just looking at the case itself what the mode of inheritance is, but the parents are unaffected, so it doesn't look autosomal dominant. So if I was presented with this case, then I might think it's autosomal recessive. So I'm going to show you how I would do a simple autosomal recessive query first. I should also point out that at the bottom of the screen here, you can see we've already loaded in the phenotypic data has already been pulled through automatically from the PhenoTips database. So this will happen automatically with your cases too. So we can see all the positive HPO terms, and you can see there's actually a longer list here than I gave in the summary. So we actually have 10 positive HPO terms and 1 no intellectual disability. Remember, in general, you don't need to put in the negative terms unless they help define the case, because obviously the case is negative for all the terms apart from the things it's positive for. But if in this case it was obviously deemed that intellectual disability might be an important feature, and this individual does not have it, so they've indicated that there's no intellectual disability. Okay. So I want to be a simple autosomal recessive, right? In that case, I expect the variant in the child to be homozygous for the alternative allele, and I expect the parents to be heterozygous at the same position. They're carriers. I'm going to leave all these fields at the default settings, because in general they work quite well, but we could change any of these. You can see for the homozygote, I'm requiring the alternative allele frequency is above 0.8. In practice, it's nearly always above 0.98. And we're requiring that the heterozygote is between 0.2 and 0.8. So nearly every heterozygote between 0.2 and 0.8 will be bona fide. Some of the ones that are outside this range might not be, but we could change these if we wanted to. And the first thing you might want to do if you're loading a new case, it makes sense if you don't know anything about the case really, or you haven't investigated it before. Let's see if there's any variants that are already described in ClinVar. So I can go here and ask for any variants that match this mode of inheritance and are already listed in ClinVar as pathogenic, likely pathogenic, or variants of uncertain significance. And we can run this query. And we get 9 variants back. So here I'm just going to show you the results menu. So this is the same as we showed before, obviously. The chromosome, the position, if it's got a DB SNP reference. The genotypes here, so you can see that the case is AA, but the parents are GA because they're heterozygotes, and you can see the gene infected. And you can see the score, and if they have any cyst or polyphen scores, they will show here. And you can see we have one called high and one called moderate, and the rest are called modified. So what do high or moderate mean? If you remember, I said you can hover over high, and it will tell you. So high is variants assumed to have disruptive impact on the protein. So these are nonsense or frame shift inducing or splice affecting, canonical splice site affecting. And moderate are variants assumed to have less impact on the protein, but affecting the protein sequence. So basically in-frame indels or non-synonymous substitutions. So these are the ones we're usually interested in. Modifier is a bit of a misnomer, and maybe we'll change it, but actually both VEP and SNP use it, and actually it means any other type of variant. So a UTR variant, a deep intronic variant, or an intergenic variant. So generally, we're not interested in modifiers. So in general, we're not interested in modifiers. Sorry about the interruption there. So generally, you will want to look at high and moderate because these are the ones that affect the protein coding sequence. So even though there's a high here, if we look across, we can see the frequency in the common population databases, and you can see that this variant is at 8% frequency in exact. If this is a rare disease, we would not accept anything that's above 2%. So this variant cannot be important, and also the variant that's moderate is at 36%. Some of the other variants haven't been described in exact, they're modifiers, but at this point, we wouldn't show a strong interest in those because it would take a long time to follow up on them, unless it happened to be very near a candidate gene. You can see the closest gene, and you had that knowledge. Normally, the internal frequency is also a very useful filter, but here, because we only have these pseudo cases, we cannot apply it today. But if you're doing your own filtering, you would always apply an allele frequency filter based upon the internal frequency as well. So I'm going to go back up. We've done one query. I could just add more things, but here I'm just going to reset. So we go back to the start. It keeps our mode of inheritance and the samples, but you see that the ClinVar is now unclicked. So I'm going to do what we would normally do since we haven't found anything ClinVar oriented. So we'll look at higher moderate variants in protein coding genes. And also, as I said, we're looking at rare diseases here, right? The platform is specifically designed for rare disease. A rare disease in Europe is one that affects less than 1 in 2,000. So we can safely filter it at 0.02, if you allow me the slight discretion that that's 1 in 2,500. But most rare diseases are 1 in 5,000 or less. Some are 1 in 100,000. So we can put 0.02 in all these boxes and be confident that we are not removing any variants that might cause a rare disease. And then we run our query again. And this time, we're getting back 10 variants. And you can see the results again. So, as a user, so this was some sort of congenital myasthenic syndrome, so this is a neuromuscular disease, right? So I'm not a neuromuscular expert. And if the person analyzing this is a research scientist, they might not be an expert in the disease that pertains to the case. In the first step, somebody can try to do the prioritization and then pass it back to the domain expert. If it's the domain expert, they'll already know which genes that they think are interesting. So I'm not an expert in this domain. So how can I get a better idea of if any of these 10 genes are of interest? So what we can do here is, I'm going down to another menu, which is the genes, disorders, and phenotypes. And as I said, we have predefined gene lists here. So the gene list that's applicable here is the muscle gene table list. And you can see if you hover on the gene list again, it's just not working just when I'm demoing, but usually a tooltip will come up here and it will show you the provenance of the gene list. So in this case, it worked for me earlier, so I know what it says. This is a list taken from the musclegenetable.fr website, where they collate genes that have been confirmed to be involved in neuromuscular diseases. And this gene list is from, I think, the end of July 2016. So it was up to date then. I think actually they have added 4 or 5 more genes, and at some point we will add them on. So you know exactly where the gene list comes from. If it's from a paper, we give the citation. So you know what these are. So if we apply the muscle gene table gene list to see if any of these genes are in that set, we get down to 1, which is like ataxin 3. So this looks quite good, apart from it's not very clear that it's a very important one, but at least we've got a gene. What can we do next? We can quickly go to OMIM and find out a bit more about ataxin 3, for example. And here you can see that this is a false positive for one clear reason, at least to me, is because ataxin 3 is implicated in autosomal dominant inheritance, and we're looking for recessive inheritance. So this is a false positive. So now we want to go back and try again. Some of you are probably screaming, but luckily I can't hear you, that it's more likely that this is a compound hit than a simple autosomal recessive. So I'm going to remove the gene list, and I'm going to click on the compound hit button. Because the compound hit is a special filter, which is described as well, then we block out some of the terms so you can't affect the results that you're given back, but the query will be a compound hit. So we're looking for 2 hits on separate chromosomes in the child, and we know they're on separate chromosomes because the parents only have 1. So if we do this, you see we have 21 hits now. 21 you would expect to have pairs, but if we look at the bottom, we can see the 1 gene here, this leukocyte, what is it? It's some sort of leukocyte immunoglobulin receptor, actually has three hits, so 1 on one chromosome and 2 on the other, so they're all possibilities. So here we have 21 possibilities, but remember here that we haven't used the muscle gene table because I unclicked it. So if we use we reapply the muscle gene table where we expect to have a gene and run the query, it will return 0. So I just did that to show that the query is working. This means that there are no muscle gene table genes affected. So what's going on here? Well, one thing we haven't tested yet, the final possibility really, so far at least, or the next possibility, is that this is a de novo. So a de novo variant will be homozygous in the parents because they don't have it, and heterozygous in the child. So I'm keeping all the filters we have, and I run the query. And here we get just one variant that's de novo, that's high or moderate, is in protein coding gene, is rare, below 0.02 or not described in the genomic databases, and is on the muscle gene table gene list. And this variant is in a gene called WIRE1. And then we can see I'm now looking at the extended results in the section above, and we can see how good the variant is, because sometimes the novel variants don't look very high quality. They can have a tendency to be false positives. However, the alternative allele frequency here is 40%. We can see that there was 41 reads, and 40% support the alternative allele, so that's very good. It's close to 0.5. We can see the parents were well covered too. They had over 30 reads and no reads supporting the alternative, so they definitely look like homozygous reference, which is what we would expect. The variant here is a glutamate to glycine change. It affects all the main transcripts of this gene. If you go to Ensemble and check, the predictive tools actually think it's likely to be damaging. So this is the actual scores from these tools. We know that already because it comes up in orange here in the background. So we know it looks like a good variant. As I said, these are just indications, but they provide a bit more belief. You can see the actual population frequencies. It's not observed in any of the population databases, and actually it's observed in 1 chromosome of our sample here. Remember, this frequency is elevated because we only have 27 samples here. In practice, if you look in the main database, this frequency would be less than 1 in 10,000. And we can see which pathways it's involved in. So here we can say this is a calcium regulation in the cardiac cell. Let's see if this works. We can look at cardiac conduction in reactome. So if you know a bit about pathways and you like to look into your pathways, you can open these tabs. Disease Card is another tool developed by associates of RD Connect, which collates a variety of data. So you can find more extensive data here sometimes. And we have links out that we're extending to other databases that might be useful. Alamut functional annotation, which is good for non-coding variants. HMT, which is good for mitochondrial variants, and Edgar. And also we have this candidate tab. So if we look very carefully here, we can see there's a tag button that I haven't described yet. And we can see it's actually been tagged once. This case was tagged on one parent, one patient. And this, when you click tag, this is to put information into the platform. It will be visible to all of the users of the platform. So if they can see this variant, they can access the tag. You say OK. OK, and this is just because it's in the playground itself. And this is the sort of information we collect. Essentially, we collect the main fields that are used that would be used for submission to ClinVar, because one of the ideas at some point down the lane is to submit all confirmed variants to ClinVar. So this is what you see. And if somebody has already tagged the variant, you can see here. And if they have comments, you can read the comments. We date stamp everything. We know who tagged it, so we have full provenance on these things. Okay. So what should we do? Because we're running a little bit short of time.
Just show other features.
Okay. So that's how to do one case. If you go, sorry, so this was okay. So if you go back to the platform website and go to try the GPAP, you will be able to download these cases. If I don't know if I have the page here. Here, download the phenotypic descriptions, and then you can try to solve another 5 cases we have here. Well, another 4 cases. So the cases 1 to 5 are all rare disease cases with different inheritance patterns, actually. So there's a clue for you. So I just want to show you a couple of more advanced features. This is a bit frustrating because it always opens a window at the top. It's the platform. Okay. So the first thing I want to show you is the matchmaking feature. And for this, I'm actually using the real platform here. I'm logged into the real platform. And if a sample is matchable, which is defined when you submit the data, this box will come up saying that you've given permission to match and you want to match. You click on the box and it automatically imports the phenotype HPO terms. Here you can see instead of numbers, they've been translated into text. And we've actually already tagged the candidate gene here. So we're interested in CHRND. At the moment, you can do internally RD Connect to RD Connect, or you can do out to Phenome Central. In the next couple of weeks, we should also have a link to Decipher. And following that, we will be linking out to other members of the Matchmaker Exchange. So I'm going to just do what you would probably do first, which is ask if there's any other samples in RD Connect that have a similar phenotypic description and have this gene tagged as a candidate gene. And we submit this. And this is going to look across all the samples. There's currently somewhere over 4,000 samples. Next week, there'll be somewhere over 5,000. And actually, we found 16 matches. And you can see the most important thing is the score here. And the way the score is calculated at the moment, according to the MME guidelines, is actually if you just have a match on the gene, that gives you a half. And any matches on the phenotype above that can add up to another half. So a perfect match would be 1. But we would not necessarily expect a perfect match because that would mean that the HPO terms that had been entered in the corresponding databases were identical. And of course, patients who have the same disease present at different stages with different symptoms. But here we have 4 matches that are above 0.5, so these are all things that look interesting. And actually this makes sense because we know this is part of a cohort where they actually all have the same variant and they have the same phenotype, more or less. So then if you found this and you were interested, you could contact the, I think if you hit submit now, no? Sorry, yes. So if you hit the contact button here, what that will do is it will send an email to yourselves, to ourselves as hosts, and to whoever owns that sample to say that we've made a match. Do you want to discuss it further, essentially? It does not show the exact data, and it does not show, I don't think it declares the exact sample. Okay, so this is a very useful feature. Another thing we've got is we've got this share and load feature. So this means you can share a query that you've performed previously with your collaborators or anybody else actually who has access to the same sample. So you don't have to tell them what filters you applied. You apply the filter and then you hit share. When you hit share, you'll get an ID, 1506, 1507 in this case. You can either copy the ID or copy the URL. And then when you want to, you can load it. And actually this will load the same thing because this is one I tried earlier. I load 1506 and actually it has worked. You just don't see it because it's put in the same data because I reloaded the same sample. So that's a very easy way to share your results. And another very important feature, specifically when you've got cohorts that have a similar disease, is you can search across all. So you can search across all samples. This is one I haven't prepared earlier. let's see if TNF alpha is a small enough gene. Actually, I can't do this. I can't do this live. Okay, so I can't do this live because I can't show you the data. But this works at the gene level. So you search across all, and this is searching across all samples that you have access to. So all your own samples and all post-embargo samples, which is most things. At the gene level, show me all heterozygous variants. And this is a very good way of checking whether any variant you found is likely to be of interest, and if anybody has a similar variant in a similar gene. And then one final feature is that we have an analysis status where you, as an analyst, can add a comment on the analysis that you have done that is recorded for later use, and again can be shared with your collaborators within SolveRD. Okay, so I wanted to show a bit more than that, but I see we're already at the maximum of the time. So now I'm happy to go back. If people want to stay connected, we will stay connected for as long as necessary, and we will take questions.
Hello everybody, my name is Ricky Joshi and I'm going to be chairing the questions here. So please write all your questions into the chat. So to start off with it, I'd just like to extend our thank yous for you joining us for the webinar today. I hope you find it very useful. And with that, we'll go ahead with the questions. So there's been a few questions that have already been written. The first one is from the TIGEM. Where they're asking, is it possible to retrieve the SNP impact on different transcripts if the reported one is not relevant?
So essentially that's what I've shown here. If you're, is my screen still shared? Yes, okay. So here you can see that RIRE1, this variant, at least in RIRE1, is hitting 4 of the transcripts. And in this case, you can see the effect impact here is moderate for all of them. What you would have to do here, if you find an interesting one, say for example, one was high and the other were moderate, you would want to go look at that transcript or consult the information here and see if it's a real transcript. What you can see here is the transcript biotype for all of these is protein coding. If the transcript biotype here was nonsense-mediated decay or non-protein coding, then you wouldn't probably be interested in that transcript if the variant impact was there.
Okay, thank you. There's a question by Giselle, who says, in the variant output tab, will the internal frequency be detailed, i.e. will we have information of homozygotes and heterozygotes?
That's a very good question, Giselle, thank you very much. And it will be detailed, but it is not detailed at the moment. So that's on our list of things to do. Unfortunately, the engineers have a long list of things to do. So once it gets high enough up the priority list, yes, we're going to show the number of homozygotes because obviously that's important for your recessive diseases. You want to know if anybody's seen any homozygotes at your particular position. So we'll separate that out.
Okay, great. If for any reason we're not asking the question that you actually mean, then please let us know, and we'll try our best to answer everything as best as we can in the chat.
Yeah, just keep asking questions at the end of the chat box as you want, and we'll get to them.
Yeah. So Dimitri asks, is it possible to upload a VCF only if you're not part of SolveRD? No, it's not possible to upload a VCF regardless of whether you're SolveRD or RD Connect. Now, the reason for this is that we always want your raw sequencing data because we put every single sample through the same analysis pipeline. And when we change pipeline, like we will do this year and change to GRCH38, we will reanalyze and revariant call every single sample in RD Connect / SolveRD at that moment. This means that we can very easily spot any artifacts that are caused by our pipeline because no pipeline is perfect. But if everything's processed through the same pipeline, they're very easy to spot. If things go through different pipelines, then you end up with batch effects and it's difficult to disentangle them. So we do not accept VCFs. If you're interested in a tool that does something similar, there's a standalone tool from the team in Marseille that will accept VCFs, a Java tool that you can download called VARAFT. So if you look for Aix-Marseille University and Christophe Bourroud, you'll find their tool, which is called VARAFT, I believe. Okay. Yeah, nice. Isabel is asking, if you have a cohort of, for example, 100 affected patients, is it possible to search for a gene which carries different potential pathogenic variants in at least 2 patients of the cohorts for a new candidate gene? I think you touched on this earlier.
Yeah, but that's a good question. So at the moment, the search across all is limited to search across all. But again, this is a very good question because this is another feature that we have on our list. And what we are going to implement is for sure search across the samples that I own. And further down the line, we might allow you to subset that, but that becomes a bit more technically difficult to implement. But say you, as Bon, have submitted 150 samples. We could let you search just across those 150 samples. And then what you would do, you would select your gene, you would select your genotype. Remember, you can select heterozygote and homozygote at the same time if you want, and you'll see them both. And I didn't show it because I can't show you the search across all results because they're in the main production platform. But for all the variants that you get, you will see how many samples have that variant. And then you can see which of your samples have that specific variant. So if you have 150 in a certain gene, you'll see that 7 have this variant, 1 has this variant, all 150 have this variant, etc.
Yep. Good. Tigem is asking a question on Matchmaker. If you want to find matches with Matchmaker, you should have identified a gene responsible or a gene candidate before, right?
Exactly. You actually have to tag your gene as a candidate for it to be absorbed into the interface automatically. However, if you do matchmaking, you can type in your gene of interest in that interface. So in our case here, because the gene of interest was already tagged, CHRND it was, not the one on screen, that automatically went into the matchmaking request. But if you hadn't tagged one, you could type in TNFA, for example, and look for a match.
Yeah. So we have a comment, RE, the sub-cohort search, that implies that you have a candidate gene already, initially.
At the moment it does. But there's no reason why at some point we will not be able to extend this. So at the moment we've limited it to 1, because obviously it's a very intensive search, right? The 1 gene, all variants across all samples. It's not normal, right? So if you do that, you're searching across 4,000 samples at the moment. The rest of the time we've been searching across 3 samples, right? So it's a very intensive search. So at the moment we don't allow you to say select 9 genes, but we could probably extend it going forward in the future and make some sort of limit. If you keep it less than 100 samples and less than 10 genes, then we allow it. This is just it's a question of the resources at the back end.
Sunita's asking, if we have only index cases, how do we channelize the compound het variants as we would get many variants?
Yeah. So obviously, if you only have the index case, it's a problem, and we can't really help you too much. But you can run the c-het analysis, and what it does is it gives you all double hits in a gene that match all your variant filters. So if you only select high and moderate and double hit across all your genes and make sure they're rare, you already get a relatively short list. Of course, the list I showed you before was about 10. I think the list in general, if you're just a single case, is probably nearer 100. Then, if you have a candidate gene list, you'll be able to get down to a shorter list. But obviously, if you want to know for sure they're c-hits, you have to examine the parents at some point.
Okay, so I have some personal messages here as well that said, can you analyze CNVs in the GPAP?
Yeah, so sorry, we didn't get, I have a slide on things to come, and maybe I should just show that very quickly, if I can get back to my menu again. Where's my PowerPoint?
Yeah.
Here? Okay. So there are things that we are in the process of developing. Okay, and particularly we're very close on these things. Very close, I mean hopefully, well, this year, and hopefully in the next 6 months. So we already know what we're doing. It's just it's quite tricky to actually implement some of these things in the platform in a way that makes it sensible for the users, but we're already working hard on this. So we're going to do CMV calls for everything, exomes and genomes. We're going to do short tandem repeat analysis for everything. We are going to display the sample QC metrics and gene coverage statistics. This will be useful because it will help you know whether your genes of interest are completely covered or not. Of course, some genes cannot be completely covered with short read technology. And sometimes an exome might be missing. And if an exome is missing, it will be useful for you to know whether that's missing for a technical reason or it's missing because it's not in the case, in which case it might be a causative variant, if you like. We're also integrating a couple of new tools for diagnostic assistance in collaboration with Mendelian.co, which is a company from the UK, and we're going to connect to the panel API from Genomics England. If you're not familiar with it, Genomics England have a list of gene panels that are very well curated by them for, I don't know, about 70 different diseases, top tier at the moment. And we're going to link directly to them. So if you're interested in a disease group that they are also already analyzing data in, we'll automatically be able to import that gene list and filter your variants to that gene list. And here at the bottom, as we say, we're always happy to receive requests for any new resources or tools to incorporate into the platform, assuming the license of whoever made the tool grants us permission, or link out to them if it doesn't. So anything that's useful to a large number of users, we will implement, and that's the way we decide on what to do.
Yeah, nice. So there's another question on sharing purposes. When a case becomes solved, do we need to do anything in the GPAP? Basically, how is it shared? Yeah,
so this is another thing I didn't have time to because we started a little bit late that we wanted to cover. But essentially, you would tag the variant. You'd tag the variant here, ideally as a candidate. You would definitely tag it in PhenoTips. As in PhenoTips, there's a field to say solved, resolved, case resolved, and then you give the gene for sure, and ideally the gene and the variant. I think within SolveRD, the agreement is that there will be the gene and the variant.
And the OMIM entry, if exists, and the OMIM entry is existing.
Okay, I think that was our last question. If anybody has any more questions, we can give you a couple of seconds to write it down quickly. If not,
so if there's not any final questions, just to tell you what I was using today, you can go here, platform.rdconnect.eu/playground. The phenotypic descriptions are shown in the red box here. And you have the username and password, which is always the same, test1234. And then you can repeat exactly what you have seen in this video, at least if you do it soon. We might change the data set in a year's time, but if you do it anytime in the next few months, you can repeat it identically. Also, if you want to find out more about anything, the SolveRD website, as mentioned by Sanya, we already have a number of RD Connect video tutorials that we've made, shorter tutorials, 3-minute, 4-minute tutorials on specific groups, on specific functions of the GPAP that will help you if you want to use certain features. And these are hosted on the RD Connect site, and actually we have an RD Connect YouTube channel as well. That's the end of what I wanted to say. So if there's no more questions.
Yeah, there's no more questions.
Any final words from Sanya?
Yes, just that we are going to publish this webinar on the SolveRD website and on our YouTube channel, so we will inform you when it is there. And thank you for attending this webinar, and I think there will be again those kind of webinars. Thank you.
Thank you very much. Thank you for all of you for giving us your time. We hope you found it useful today. And if you do have any questions, you can always email help@rdconnect.eu or platform@rdconnect.eu. Those emails are on the platform homepage.
Okay. Thank you to you too.
Thank you. Bye-bye.
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 Bye-bye.